My name is Neil Watson. I'm the chair of psychology here at SFU and a professor of neuroscience. It's my pleasure to welcome you to the third installment in the President's Dream Colloquium for 2017, Understanding Medical Cannabis. We have, as many of you will know, 6 outstanding speakers lined up for this colloquium. Today, we're very pleased to be welcoming Dr. Francesca Filbey from Texas, and I'll come back to her in a moment. I want to point out that in 2 weeks' time, we have a colloquium with Dr. Mark Ware, who will be speaking about pain management aspects of cannabis use. 2 weeks following that, the Honourable Anne McClellan, former Deputy Prime Minister of Canada and the chair of the committee with respect to legalization of marijuana in Canada, will be speaking at Harbour Centre, not here, on those issues. And our final guest will be Dr. Vogue Kilmer, also speaking on the topic of legalization from a more academic perspective, also downtown. I also want to draw your attention to some resources available to you. There's a website associated with this colloquium series that I encourage you to visit. As you can see at the top here, sfu.ca/medical marijuana. It'll give you information about the public lectures. These are being webcast and also live streamed, so you'll have access to this material later should you desire to come back and revisit some of the topics. And lastly, after today's presentation, there's a reception outside here with food and drink. I encourage you to stay after and mingle with faculty, staff, and students, and of course, Dr. Filbey, who will be here to talk some more informally about her work. We've been lucky to receive sponsorship from a variety of organizations, both on campus and off. The President's Office, of course, is the main impetus behind the President's Dream Colloquium series. The SFU Faculty of Graduate Studies and Postdoctoral Fellows, Faculty of Science, Kwantlen Polytechnic University, and the Natural Sciences and Engineering Research Council of Canada, who of course are one of the sources of much of the scientific funding for research on campus here, and many industrial partners as well in the private sector, too many for me to name off here, but they're listed on this slide, and we thank them very much for their generous support of this series. So it's my pleasure to today introduce Dr. Francesca Filbey. Dr. Filbey comes to us today from the University of Texas at Dallas, where she's currently associate professor and director of cognitive neuroscience research of addictive disorders at the Center for Brain Health in the School of Behavioral and Brain Sciences there. She's also an adjunct associate professor of psychiatry at the University of Texas Southwestern Medical Center and an honorary researcher in the Institute of Psychiatry at King's College London. Following a master's degree at the University of Texas, Dr. Filbey completed a PhD in experimental neuropsychology at King's College under the supervision of Professors Sir Robin Murray and Robin Morris, followed by a postdoc in neuroimaging in 2005 at the NIH in Bethesda with Robert Cohen. Dr. Filbey's program of research is devoted to understanding the biobehavioral mechanisms of addictive disorders, and in this pursuit, she melds together the perspectives and techniques of cognitive neuroscience, neuroimaging, genetics, neuropharmacology, psychology, and psychiatry, so casting the net very, very broadly through neuroscience to inform her work. This uses, her work is really at the technological cutting edge, taking advantage of brain imaging technologies, including MRI, electrophysiology, functional infrared imaging technology, which is super cool, and powerful genomic approaches like genome-wide association studies. The overarching goal of the work is to understand and identify markers and mechanisms of addiction and thereby improve both identification of at-risk individuals and help develop effective intervention strategies. Her record of accomplishment in this regard is really extraordinary. She's the author of more than 70 journal articles in many of the very best journals in our field. She's generated a number of contributed chapters, an edited book, has been awarded the Burt Moore Endowed Chair in Brain Health at the University of Texas, elected to several editorial and professional boards, recognized by a variety of prizes and awards from scholarly organizations in the domains of neuroscience and addiction. The title of Dr. Filbey's talk today is Marijuana in the Brain, From Addiction to Neuroscience. And would you please join me in welcoming Francesca Filbey to the podium.
Okay, I think everyone can hear me. Thank you, everybody, for being here. It's a pleasure to be here. I always take the opportunity to come back to Vancouver, beautiful city, and I'm even more excited to talk to you about what we know about marijuana in the brain, both from the literature and from my own research. I've been studying marijuana specifically for the last 10 years as funded by the National Institute of Health. And my overall or overarching goal really is to determine who are those individuals who are highly susceptible to marijuana's effects, who go on to have problems with it. We all know that by and large, majority of the population who use marijuana come out perfectly fine. But wouldn't it be much better if we knew your susceptibility is, that we knew who those individuals are who may be taking a chance of ending up with problems related to their use so that they can have or make better informed decisions. So that's really the goal of my research. And along the way, we've also been able to find discover what it is that happens to individuals who use marijuana, with the objective, again, of determining the relationship between marijuana and the brain. And along the way, what I've found is that we really know very little about marijuana's effects on the brain, which is really surprising to me, because marijuana has been around for a long time. And in fact, in 2008, the Journal of Experimental Botany published this paper showing that the oldest evidence of marijuana use was 2,700 years old, from 2,700 years ago. This is as you can see here, this is the remains of a medicinal man from that period of time, and they found him buried with about 2 pounds of marijuana. And so it's been around for a long time, but we still really know very little about it. And what I mean by the fact that we know very little about it, we know little about its long-term effects. We especially know little about it relative to other substances, whether it be alcohol, cocaine, and tobacco, which I think is very surprising given that marijuana is the most prevalent illicit drug. Illicit in terms of when these statistics were collected. In about 2010, in America, about 11.5% of individuals over the age of 12 reported having used marijuana. As you can see here, these are the number of illicit drug users in millions. The top is the cumulative number of those who use illicit drugs. And then here's marijuana use at about 18.9 million, which is extremely large relative to the other types of illicit substances. Look at cocaine here at 1.6 million. It pales in comparison relative to the amount of marijuana users. About 10% of these individuals who use marijuana, as I mentioned, end up with problems related to their marijuana use. And what I mean by that is that they end up not being able to stop even though they want to. We refer to these individuals as having cannabis use disorders, or what was previously referred to as having abuse or dependence. So these are the individuals who despite having problems either socially, occupationally, financially, or just wanting to cut down, have difficulties doing so. So the 10% of those individuals, you can see in this slide here, or in this graph here, that number of individuals, or the rate of treatment-seeking individuals. And I'd like you to focus on the two highest bars here. The green one here shows you number of treatment-seeking individuals who primarily go in for alcohol use, which shows here the highest rate of treatment-seeking individuals. So that's alcohol. And the green indicates the age range, which I don't know if you can see it here, but these are individuals who are over the age of 26. So the highest number of treatment-seeking individuals are those above the age of 26, and they do so for alcohol. Now look at the second one here, very close second in blue. These are individuals who, whose number one reason for seeking treatment is for marijuana. And the blue indicates the age of those individuals, and these are individuals between the ages of 12 to 17 years of age. So that is a concern, again, because the number of individuals who end up needing help for their marijuana use are about 10% of the population, and it looks like the greatest rate of those treatment-seeking individuals are 12 to 17 years of age in adolescence. It's even more a greater concern because treatment these days are largely ineffective. The rate of relapse, especially after about a year of following treatment, is as high as 70%. So even though these individuals are seeking treatment, they may not be effective. So they may end up having lifelong challenges ahead, which is really problematic if you are seeking treatment already at such a young age. Another concern for not knowing what the real relationship is between marijuana and the brain is that there's a changing marijuana use landscape, and we are all in the midst of it now, and you are all highly sensitive to that, being here in Vancouver. We are experiencing the same changes in the US, where shown here in light green, 4 states have already legalized recreational use. So that's Washington, Oregon, Colorado, Alaska, and this is in addition to the District of Columbia, and then 16 who have legalized medicinal use of marijuana. Since Colorado legalized recreational use, they've also seen an increase or a significant increase in recreational sales. I'll point you to the last 2 points here, which shows the jump in sales from in December of 2014 to the first month of 2015, where it jumped significantly. But if you compare the what it was the year previous to, it was at 12%, or the increase was 12 times the amount reported for recreational marijuana use sales. Another evidence for the changing landscape is just a change in attitudes towards legalization of marijuana. As you can see in light green here, these are individuals who said that they oppose legalization, whereas the green line are those who said that they do support legalization. And you can really see the shift in the trends here, starting from about 2011, where where there's a real sharp switch in attitudes towards legalization. So much so that where I'm from, Texas, have also already begun discussions on changes in legalization. And in fact, in Texas, medicinal use for marijuana, specifically for pediatric epilepsy, is approved. This is a report that the National Institute of Drug Abuse has published, and it talks about what we know in terms of the group of individuals that are most at risk for these long-term effects of marijuana use. And this report comes from several years of research into what we know in terms of outcomes in adolescents who use marijuana. And what they report is that 78% individuals who use marijuana initiate their regular marijuana use during these highly critical neurodevelopmental period. It also shows that these individuals' risk of harm, that when it goes down, their rate of use goes up. So there is definitely an inverse relationship between those two. And that every day in the US, about 3,000 teens use marijuana for the first time. So I think this is really important to remember because the adolescent brain goes through a very rapid period of change. This goes along with brain maturation. We see connectivity in the brain during that period of time sharply maturing and developing. Excess gray matter is pruned, and these connections between regions become stronger. And so any exposure to any type of substances, including marijuana, during that period of time could lead to long-term irreversible effects. There have been some studies that have reported a drop in IQ points decades later following adolescent use, and heavy marijuana use has been linked to poor educational and social outcomes. So that's what we know so far in terms of marijuana use, the rates, the prevalence, and also the potential of harm in some high-risk individuals. So what do we know? What do we know about what marijuana really does to the brain? Well, we know that cannabis, the plant, has psychoactive ingredients. Right now, there are about 483 identifiable chemical compounds. 113 of them are considered cannabinoids. So cannabinoids are chemical compounds that bind to specific receptors in the brain called cannabinoid receptors. The kinds or the most well-known cannabinoids in the cannabis plant are tetrahydrocannabinol, cannabidiol, and cannabinol. These are just examples that I provided there, but there are, as you can see, a lot more. So I mentioned that these cannabinoids bind or attach to certain or specific receptors that exist in the brain, and we refer to them as CB1 for cannabinoid receptor 1 or CB2 for cannabinoid receptor 2. I don't know if you can see here, but this illustration shows you the location of those cannabinoid receptors. CB1 is in light green, which you can see is predominantly in the brain. And then CB2 in blue here shows that it's predominantly in peripheral organs, especially within the immune system. THC specifically attaches itself or binds to CB1 receptors. Cannabidiol has indirect effects on both CB1 and CB2 receptors, and cannabinol have specific affinity towards CB2 receptors. So what happens when THC is, when the brain is exposed to THC? What are the events that follow exposure to THC in these receptors? So I'll just take you through that. What happens is that these illustrated here in green is the THC molecule that binds to a receptor, which then leads to a release of neurotransmitters. So neurotransmitters, which are illustrated in these circles here, are basically how our neurons communicate. Okay, so here's one neuron which sends the signals, and here's another neuron that receives those neurotransmitters. Neurotransmitters are also chemical compounds that carry that information. So when THC binds to the receptors, causes the release of neurotransmitters that then sends out information and causes nearby neurons, nearby brain cells, to respond by releasing cannabinoids. So the reason why we have cannabinoids or CB1 receptors in the brain is because we actually do have cannabinoids that naturally occur in our brain. So when those cannabinoids that naturally occur in our brain are released because THC has induced their release indirectly, the cannabinoids go back to the original receptor that released the neurotransmitter in order to try to diminish the action of that neuron. So basically, what this all means is that the role of the cannabinoids is to modulate the effect of these neurons. You can think of it sort of as a dimmer switch or a volume control. So that's essentially what happens when THC is in our system. So you might be wondering, well, why do we have cannabinoids? Why do we have naturally occurring cannabinoids? What is this system for? We call this system the endocannabinoid system, and it's because its main function is to regulate various physiological processes. I already described how it dims down or turns down the action of other neurons. And because it's all over the brain, as you saw from the previous slide, it's involved in various functions, such as higher order cognitive functions. And what I mean by that are the kinds of cognitive functions that we need to guide our behavior through learning and memory, through planning, decision making. It also controls functions like pain and appetite, our immune system response, mood, as well as reward and motivation. So there are three classes of cannabinoids. We have endocannabinoids, and these are the ones that are naturally occurring. Anandamide and 2-arachidonylglycerol, or 2-AG for short, are the two most common ones in our brain. Phytocannabinoids are the ones that are plant-derived, and I already mentioned that the most common ones are THC, CBD, or CBN. And then there are also synthetic cannabinoids, which are laboratory derived. Some examples of those are marinol and K2 or spice. So where are these receptors in the brain that THC binds to that then causes actions within? This slide shows you the areas of greatest concentration of CB1 receptors. And by looking at this, you can see how it's pretty ubiquitous. It's almost the entire brain. But I'll point to some of these for you so that you can see the regions that underlie some of the behaviors that follow marijuana use. So one of the greatest regions is the cerebral cortex. This is the area for higher cognitive function. So marijuana or THC affects the cerebral cortex functioning through impairment in learning and memory, and also disinhibition. Another area is the hypothalamus, which is, again, rich in CB1 receptors. So when THC binds to brain cells in that area, it leads to increases in appetite. The hippocampus here is important for learning memory. And also, not illustrated here, but the amygdala close to the hippocampus is important for stress and emotion. And then other areas here which are important for movement, nausea, also important for sleeping behavior. And then the spinal cord is important for sensation, which includes pain. I'm not going to go into this too much since I already mentioned or briefly introduced you to how THC leads to a cascade of events. But as I mentioned, what it does after it induces a release of neurotransmitters and cannabinoids are then released because of the neurotransmitter action, is that it tries to, again, modulate or dampen down the what we call presynaptic receptors, or the neurons that sent out those signals. We call this retrograde signaling. So in this example here, if the nearby neuron happens to be one that induces excitability, one that increases brain activity, it might suppress that or inhibit some of that action. If the nearby neuron happened to be one that is inhibitory or dampens down action, then it might suppress that, and then leading to higher excitability of the system. And so because with THC, with exposure of THC into the system, it's possible that there could be complex interactions because of the nearby neurons, because there's no specificity, and the actions depend solely on what the neurons are that surrounds that presynaptic, or that neuron that releases the cannabinoids. And what's even more important to understand here is that it could flood the system with cannabinoids that sort of just stay within the extracellular space that continue then to exert their efforts. And so the brain is a very fascinating organ in that it adapts to these changes in its environment. So with time or with repeated flooding of cannabinoids, it might adapt to this new environment by decreasing the number of receptors that it has to try to maintain an equilibrium. And the reason that I mention that is because that really leads to differences in short and long-term changes following marijuana exposure. Okay, so we refer to the short effects as acute effects, and the long-term effects chronic effects. And the chronic effects may be because of those processes that I just described to you, the way that the brain tries to maintain or go back to some homeostasis, some balance in the system. So this part of the image here, part of the slide, shows you the changes that happens in these brain regions in terms of chemical availability or chemical synthesis. And this shows you what happens to it following long-term use. And this comes from a review paper that came out very recently. So I'd like to just highlight a very relevant neurotransmitter that's illustrated here, and that is dopamine. Dopamine is a primary neurotransmitter within the reward system, and it is also modulated by cannabinoids, so by THC. And here it shows you an arrow illustrating that there's an increase in dopamine levels during acute exposure or short-term effects of marijuana. But if you look here to when following long-term or chronic exposure, you see that the brain tries to adapt to this chronic flooding of cannabinoids in the brain by decreasing levels of dopamine. We've also, in the animal literature, have found neural changes in the brain. Here in this study that came out, what they did is that they exposed rats to synthetic cannabinoids to try to emulate what the brain might look like following exposure. And they use two different compounds. So the top half of the brain shows you one type, and then the bottom shows you another. And then these different images shows you each time period following initial exposure. So if you follow me here, this is this is the baseline period where they haven't been exposed yet. And this is 3 days, 7 days, 14 days, and 21 days following exposure. And it's difficult to really get a summary here. But if you just focus on the red areas here, that shows you where the receptors are in the brain, in the binding, and you'll see that, if you follow my pointer here, that from baseline to about 21 days, that there's a real shift, there's a decrease in binding or receptor availability from baseline to 21 days following exposure. These graphs better illustrate that for each of those regions. This is for the cerebellum, hippocampus, and striatum for each of the compounds, which shows a trend, a decreasing trend from the first day of treatment to after the 21 days of continued treatment of marijuana. Similar changes have been found in human neuroanatomy. This is a figure from a review paper that came out recently. What this paper did was it combined all of the regions, brain regions that have been reported to show changes in marijuana users. The top left here shows changes in areas in blue. And then here's an image showing where the CB1 receptors are. And then to make it easy, the authors also overlapped or tried to merge together areas that showed alterations where there are CB1 receptors. So as you can see here in red, cerebellum is predominant area where they found that, and the hippocampus as well, right here. This area in pink, or this image, shows areas in pink where there are no overlap. There is no overlap between the areas of alterations with CB1 receptor density. So the bottom line here for this image, or the take home, is that the areas that show changes don't necessarily have to be areas that are dense with CB1 receptors in humans, suggesting that there may be some downstream or indirect effects of THC in other areas that are not directly targeted by THC. This graph here to the right is again from this review paper, and it shows you what those changes in those regions may be modulated by. The first one shows you its relationship to dosage, so dose effects. This second one shows you its relationship to duration of use. And then the last one shows you whether age of onset has anything to do with those alterations. On the y-axis here are percent or number of studies that have found associations between those 3 cannabis use measures and changes in the brain. The blue shows you a negative effect, and the red shows you positive effects. So if you look at this, most of it looks, it's very difficult to synthesize because it's very inconsistent, right? You see both red and both blue, and if I really had to look at it, I would say that there's really not much of an association. There's really, like, look at duration of use, for instance. There's only really very few percent of research that have found a significant association with brain alterations and duration of use, and same with age of onset. And in terms of dosage, there may be a little bit more there, but by and large, very inconsistent findings. So what that really says is that not only do we know a little about the effects of marijuana use on the brain, at least the long-term effects, but in humans, it's largely inconsistent. And that's because the way that we have studied marijuana users have been impeded by just the difficulty in studying this population, but also because of very non-standardized or inconsistent methodology. One of which is just in the way that we sample marijuana users. Who are these individuals? Some sample individuals who have barely used marijuana. Some sample individuals who report heavier use. So we really just need to establish more standardized ways of studying these effects. And so, I'm going to shift gears now and talk about my approach and describe to you the findings from my studies. So how have I tackled this question? So again, with the goal of identifying individuals who might be at heightened risk for having problems with their marijuana use, I wanted to look at heavy using marijuana individuals. So these are not individuals who just use marijuana socially or recreationally. These are individuals who have used marijuana consistently over a long period of time. In this slide, I'm showing you the actual data or the actual characteristics of the participants I use in my studies, and that the findings to follow are from. They are in 2 columns here because they came from 2 different locations. These individuals were recruited from New Mexico, while these individuals were recruited from Dallas, where I am now. We had 148 marijuana users recruited from New Mexico and 96 from Dallas. Even though they were from 2 separate cities, the criteria for participation in the studies were exactly the same. These are long-term marijuana users. They're mostly males, and in fact, this should be greater. This should also be 50, around 50. And look at their mean use or mean days of use per week. It's nearly daily. And then the number of times a day that they use, these are separate occasions of use. It's around 4 for both groups. And again, they've been using regularly for several years, 6 years for this group of individuals, and about almost 10 years for those for the ones I recruited in Dallas. These individuals in blue are non-using individuals, and these are naive, marijuana naive individuals that I use as a point of comparison. The techniques that I use in order to measure effects on the brain are based on magnetic resonance imaging, or MRI. And I will go through each of the findings based on the 3 different methods that I used. But very briefly, I used structural MRI to look at the anatomy of the brain, diffusion tensor imaging, which looks at the white matter tracks, the fibers that connect the gray matter, or the brain regions. And then I also used functional MRI, which looks at brain activation when individuals are doing something in the scanner. I'm going to just give you a brief orientation as to how to interpret fMRI maps or images if you are unfamiliar with them. But throughout my presentation, I'll be showing you images like this, which are brain images. And the colored areas we refer to as blobs, but they really reflect activity in the brain. Typically, the blue blobs show some deactivation where the brain is not responding, whereas red indicates higher activation, so when those brain areas are engaged during a specific task. So let me start with what we have found using structural MRI. This graph shows you 2 areas of the brain, the hippocampus, which is shown here in yellow, and the amygdala shown here in blue. So again, the hippocampus is important for memory, and amygdala is important as it regulates stress response. And what you see here in columns are left hippocampus, right hippocampus, left amygdala, and right amygdala. And the white bar shows you healthy controls, and then the black bar shows you the results for cannabis users. And what we're showing here are volumes of those regions. So again, if you look closely here for the left hippocampus, you see that in the black bar, the cannabis users have smaller hippocampal volumes. Both left and right are smaller than the healthy controls, the non-using controls, and also the left amygdala. The right amygdala, although it shows it's a little bit less, was not significantly different. But the left amygdala did seem to indicate smaller volume in cannabis users than the non-using controls. The same was found in the orbitofrontal cortex. These are the areas, or this is the area of the orbitofrontal cortex that seem to indicate decreased volume in these long-term marijuana users, which is really interesting because the orbitofrontal cortex is a part of the reward system, the brain's reward system, which is important for sustaining us as organisms. It's important for seeking out things to keep us alive. Let me see if I have, no. So for instance, the orbitofrontal cortex is right here. The orbitofrontal cortex, the role of the orbitofrontal cortex is for adaptive learning. It helps us learn what it is that is good for us, that we should continue to seek out, that then would motivate us to continue to seek. So the reward system, again, is important because not only does it help us survive, but it also has been shown to be important for social companionship. And the primary neurotransmitter, the primary molecule for the reward system is dopamine. So the next slides I'm going to show you will talk about our findings in terms of using the diffusion tensor imaging, or DTI technique. And our measure of of the integrity of these white matter tracts is through an index that we refer to as fractional. Actually, that should say fractional anisotropy, which is how we get FA. So high FA, or fractional anisotropy, basically says that the movement of water molecules within the white matter tract of the brain is very uniform, okay? If you have low FA, or low fractional anisotropy, it means that there's not much directionality in the movement of the water molecules, which could suggest some decrease in the integrity of the white matter tracts. Okay, so if you just imagine that that's a white matter tract, the movement of the molecules is more diffused. So going back to the orbitofrontal cortex, we wanted to find out what this means given the lower volume that we found in the marijuana users. And what we also found was that the connectivity in the marijuana users was decreased. And that was in these tracts shown in red that we refer to as the forceps minor. The blue areas here are the regions of the orbitofrontal cortex that showed a decrease. And so with decreased volume, we also found lower fractional anisotropy. And so what I wanted to do then was determine how that decrease in fractional anisotropy relates to some measure of cannabis use. And so what I looked at was continued years of use, or years of continued use. And you can see the relationship here is that there is in fact an initial increase following initiation of use. So up till about the 5th or 6th year of continued use, there is an increase in white matter integrity, but then following long-term use, there is that decrease in FA. Again, maybe hinting at some of the long-term neural adaptations that we've found before in other research. There's also another way of looking at connectivity between regions. So because we found that structurally the white matter integrity seems to be lower, what does that mean in terms of its function? And we can look at function using a technique called resting state functional connectivity. So functional connectivity here is basically how these two brain areas activate together, whether their response or their activation is highly correlated over time, whether they have synchronized activation. So shown here again are the forceps minor that I showed you already had decreased white matter integrity. But then when we looked at functional connectivity, so whether areas of the brain might be highly synchronized, so in this case, the orbitofrontal cortex, despite the lower white matter integrity or structural connectivity, what we found in marijuana users is that it was actually greater in the users and heavy users relative to non-using individuals. So our interpretation of that is that it might be, or two things. It might be that because there's greater connectivity in these regions, there is no need for the regions be large, right? Because it's the connectivity, it's the communication between these two regions that really matter. And that's, in fact, how our brain matures, by increasing connectivity or communication between them. It's also possible that the decrease in the size of the regions that's led to the increased functional connectivity. It might be some kind of compensation, perhaps because of the loss in volume. We wouldn't be able to answer that question because this wasn't a study where we followed individuals to see what happened first, but it's definitely an interesting one and hopefully one that we can answer at some point. So now I'm going to talk to you about our findings using fMRI, again, which shows you how the brain functions in these group of individuals. Before we do that, let me just highlight the conclusions from those set of studies, and that is that there are in fact gray and white matter alterations, both structurally and functionally, and particularly in the orbitofrontal cortex of the brain, again, an area of the brain important for reward. So why is it important to look at reward? It's important to look at reward for several reasons. First, reward mechanisms or reward function lies in areas that have a high amount of CB1 receptors, again targeted by THC. Looking at reward is also important because it's changes in reward function that drives problems with substance use, including marijuana use. And in fact, changes in reward sensitivity has been considered a primary symptom in addiction, and that is measured in terms of response to marijuana cues, which elicits this high desire to use marijuana, referred to as craving. So just to give you a sense of what craving might be like, to a lot of people, being exposed to this might make them want to seek out or find their nearest Starbucks to drink a pot of coffee. So it's this high intense desire or this drive to seek out a substance following exposure to an object or a cue that has been conditioned to the pleasurable or rewarding effects of that substance. So how did we do this in the studies to look at craving mechanisms or this response to cues in marijuana users? What we did is we presented individuals with tactile stimuli, and in this case we used a marijuana pipe. We exposed them to the marijuana pipe, and then we asked them to rate their level of urge to use marijuana while they were in the scanner. And then we compare that to a neutral object like a pencil. So again, these are the images that I'm going to be showing you, but I'm going to point you to the areas of interest so you don't have to worry so much about the overwhelming amount of red in this slide. But I want to show you, first of all, that there are, that the response, the activation in response to the presentation of a marijuana cue was pretty widespread. These are slices all the way across the entire brain. So as you can see, again, shown in red, that there's numerous areas where that responded greater to the marijuana pipe relative to the pencil. I'm going to show you or point you to the areas here because that area is the orbitofrontal cortex. So again, important area in that drives reward processes. So what that shows you is that when marijuana users are given to them a marijuana pipe, and in fact, not only are they holding it, but they can see it because we place a mirror above their eyes so that they can view their own hands looking at the pipe. What we find is an increased response in several areas of the brain that includes the orbitofrontal cortex. What's even more interesting here is that we correlated that response to cannabis use behaviors, one of which is this questionnaire called the Marijuana Problem Scale. This asks questions about anything in their life that might be related, might be challenges surrounding their marijuana use, and we also looked at age of onset. So here, the red colored areas are the areas that were associated, so that responded, that had greater response with greater number of problems reported in this scale. As you can see, there's really not much more in the brain that was associated with problems with marijuana, except for this region here. And lo and behold, it's also the orbitofrontal cortex. So to conclude those studies, we find that there is greater reward response to marijuana cues in the marijuana users, that it was related to marijuana reported problems, such that those that had greater problems related to marijuana use had greater response in the orbitofrontal cortex. But the real question is, okay, so you've shown that marijuana use has resulted in this greater response to a marijuana pipe, but wouldn't that be the case for everyone because you're comparing it with a pencil, right? Of course, we're going to respond more just because, it's much more interesting than a pencil. And this concept of hijacking the brain, really what that is referring to is whether it's taking away the normal processes of the brain. So in terms of the reward system, is it taking away the normal process of the reward system, which is in seeking out naturally rewarding stimuli, such as food? And so we looked at that in the follow-up study, where we looked not only at the response to a marijuana cue, but we also looked at its response to the cue relative to a natural reward. So what's a natural reward? Before I tell you what the natural reward is, another modification that we did was that we didn't just show them a pipe, which was in the previous study, similar across all participants. In this version, we asked them, we presented to them their chosen marijuana cue. So a cue that was actually that they were actually familiar with or that they use most often. So we presented them with whatever it was that they liked. And then in terms of natural rewards, we wanted to find something that should be highly familiar to everyone, but it's also naturally occurring. So we chose fruit. And just like individualizing the marijuana cues, we individualize their natural reward cues. So we gave them an option in terms of what fruit they wanted to be exposed to in the scanner. We gave them the option of an apple or a banana or an orange or grapes. So the question here is, does marijuana lead to greater response in these same areas of the brain much more than it does for natural rewards like fruit? Let me first start with what we should have already answered before, but just to check that we replicate our previous findings, and in fact we do. You can see again that in marijuana users relative to non-users that there is greater activation in the brain, pretty widespread as you can tell by the amount of red on that slide relative to the pencil. So we already know that. In non-users, there were also areas of the brain that were greater to the pipe relative to pencil, but not so much, right? And in fact, there's nothing in the way of the orbitofrontal cortex, which you could see here is still present in the marijuana users. All right, I'm going to go over what we found when we looked at response in the brain that's greater for fruit relative to pencil. So what would you expect? Here you would expect almost the same, right? Because fruit is naturally rewarding. It should also trigger responses in the brain because it's a natural reward. Yeah.
This is within subject?
This is marijuana user versus non-user.
Pipe versus fruit.
Oh, yes, these. So everybody got the exact same set of cues. They were shown a pipe, pencil, and a fruit. And yes, in marijuana users, relative to non-users, we still found a lot of red, right? A lot of response across different brain regions that were greater for fruit relative to the pencil. Again, you're saying, well, it's the pencil, so of course we expect that. And we also find the same in non-users. So again, we expect this, so we didn't find anything too strange there. So, but the real question is pipe versus fruit. So what do you see here? You see greater response to the pipe in the marijuana users relative to fruit, but in normal controls, you don't. And you should be familiar with this area already here. Again, that's the orbitofrontal cortex. So what does that tell us? Well, it tells us that marijuana users are definitely responding to the cannabis or the marijuana cues, that it is greater than fruit. But we also know that they still respond to the fruit greater than pencil. And I should mention here that when I did the reverse, when I looked at fruit greater than pipe, and I apologize that I didn't add this slide here, there were also areas that were greater for fruit relative to pipe, but it did not include the orbitofrontal cortex. So what does that say? It says that there are greater reward, we see continued greater reward response to marijuana cues in the marijuana users, but it's not so much that they're not responding to natural rewards. They are still responding to natural rewards, but the response is a little bit different. So this supports a model of addiction called incentive sensitization, where more potent substances change the way that the reward system responds, but it's not so much that they were deficient in reward functioning from the very beginning and that they needed these highly potent substances. So what we're seeing here is a potential mechanism by which those who use marijuana may end up having problems related to their marijuana use. So what about motivation in the reward circuit? Because motivation is also one of the processes that is important in this reward system. We looked at, again, using fMRI, we looked at what this neural response in the brain might be to motivation to financial gain relative to financial loss. So we are motivated not only by the prospect of something positive, right, like a reward, but we're also motivated by the prospect of avoiding punishment or avoiding some kind of aversive condition. So how we did this in marijuana users was we scanned them while they were performing a task called the monetary incentive delay task. This is a very well-utilized cognitive task, and we did this in the scanner. And it's basically a test of how much individuals anticipated a potential gain, monetary gain, or anticipated a potential loss. So what they did in the scanner was they were presented with these visual presentations that indicated whether they have a chance to gain money, so shown with a plus side, or lose money indicated by a minus sign. Following that signal, they were then shown a gray target that they have to respond to. They are told that if they are able to press a button while that gray target is up, that they actually won that money. If they are not able to, then they actually lose that money in the loss trials. And then, in fact, they are told whether they were successful or not, and they are actually shown their cumulative amount of earnings, and they are actually given that amount of money in cash just to increase motivation throughout the task. And so what we were interested in is this time period when they are told whether they could potentially win or whether they could potentially lose. What is it that drives motivation in these individuals? Okay, so again, I'm going to show you findings based on marijuana, the marijuana using group, and then also the non-using group. So this shows you the difference between a possible gain, a win, versus a possible loss. Again, this is their anticipation during gain versus loss. And what do you see? Again, the orbitofrontal cortex seems to respond greater for a possible win rather than a loss. Interestingly, the non-users, they were not responsive at all to the gain, or at least the difference between potential win and potential loss was not that different. Same with when we looked at loss, what's greater during a potential loss versus a potential gain. The marijuana users, nothing really. So when there was some type of negative reinforced motivation, it didn't seem to trigger or elicit a response in the brain of marijuana users, which is the inverse in the non-users. So there seems to be an interaction here between the groups and the type of incentive that we present them, where in the non-users, they seem to be more motivated by avoiding harm, by negative reinforcement, whereas marijuana users seem to be motivated, or at least areas of the brain responded more to a potential gain. Okay. I'm going to this is a very new study that we just started, so I'm going to just give you a very brief summary of what we found. We also looked at learning and memory using a task called the serial reaction time task, and also collected EEG data on them. But basically what they did was that within each block, shown here in squares, they learned a certain sequence. So they learned a sequence of buttons. You guys remember the Simon task, or not the Simon task, the Simon toy where the lights lit up and you have to press them in the order that they lit up in? It's basically like that, where they're shown a sequence of areas on the screen, and then they have to remember the direction that they appeared in. And it's the same set of sequences over and over again. And what we wanted to see is how quickly are they able to learn that sequence. They were shown that in 5 times. And then after the 5th one, they were shown a new set of sequence, and then tested again to see if maybe some of that skill transferred over to the next one. In this graph, you'll see their performance plotted across all of those blocks, across all of the times that they were supposed to learn the sequence. If you are learning, you should be getting faster and faster. So this should be decreasing, should be a downward slope. But it's not. It's actually pretty random. And then when we looked at their EEG, again, across the blocks, we found something interesting looking at their alpha and their beta responses. Alpha waves, brain waves in the EEG, are usually present during rest, during drowsiness. But beta waves usually reflect periods of being awake or being alert. And so interestingly, in this group of individuals, we see a lot more alpha waves, suggesting that they might not be fully engaged in the task while we were collecting the data. And in fact, the alertness or awake signals, as reflected by the beta waves, seem to be pretty flat all the way across. So this might be why they aren't demonstrating learning across those learning trials. So this impaired learning might be associated with low alertness in marijuana users. So again, with the goal of determining or identifying individuals who are at risk, so that we can identify who they are early on, and so that they can also have early targeted intervention if they need it, we look to see what these potential modulators are, or potential factors that increase susceptibility. Onset of use is one particularly important factor, because those who have had earlier onsets of substance use show greater effects. And in fact, that's what we found, was that when we looked at age of onset of regular cannabis use, we found the areas of the brain for control, for inhibitory control, had less activation. And this was during the exposure to the cues. Early childhood stress has also been associated with heavier substance use, or with substance use, because of the interaction between the stress and the reward systems of the brain, and how, again, early life alterations might lead to these long-term consequences. What this image illustrates are the amount of the dopamine signals in the brain in animals who were raised in a stressful environment, which is basically in isolation, versus those who were not in a stressful environment, which is those who were socially housed. As you can see, that there's greater dopamine receptors in those areas. So this is just the foundation for looking at what early childhood stress how that might affect cannabis use in these individuals. And what we did was we conducted a study in 12 to 17-year-old individuals, and asked them whether they ever consumed any of these substances. Here's for alcohol and marijuana. And what we did was we correlated it with the amount of childhood adversities that they had, or that they reported. Here, childhood adversities were really significant adversities in life, like a death of a loved one, or a natural disaster, things like that, or significant hospitalization. And as you can see by these red arrows, there's a high correlation of the amount of early childhood adversities reported and the engagement in alcohol and as well marijuana use. So there's some suggestion that there could be a relationship there. Stress is also acute stress specifically could also lead to changes in the reward system function. And so what we did is we provided a stressor while we scanned individuals. What's really stressful for a lot of us is performing arithmetic tasks within a specific amount of time. So we asked individuals to perform very difficult arithmetic problems. These were calibrated based on their skill level. But not only that, but we gave them a certain amount of time that they needed to complete the equation in. And for the social stressor factors, because we wanted to incorporate that as well, we showed their performance relative to the performance of an average participant, so if this was the individual, the bar graph here shows their performance, which is less than that of the average user. In addition to that, we also had the research assistant who's collecting this data closely monitor them, so always reminding them that they're being monitored throughout and that it's really important that they did the best that they can. So needless to say, it was a very stressful task for everyone. And in fact, again, this is their brain when they were engaged in this stressful task. And we found areas, as expected, in the amygdala, but also, again, in the orbitofrontal cortex. Response to stress was then correlated with the amount of problems that they reported related to marijuana use. And what this shows you is that in these areas of the brain, we show that greater response during stress, during that difficult or stressful task, was led to greater problems, or was correlated with greater marijuana problems. Due to time, I'm just going to summarize these last few slides. That basically suggests we've shown that CB1 receptors are definitely important in marijuana users, and so it's logical to speculate that genes that regulate or code the amount of receptors that we have in our brain could make us or some individuals more susceptible to marijuana's effects than others. So it's possible that that genotype could also put one at risk. And in fact, looking at two genes that code for how the endocannabinoid system functions do show an effect. These are the cannabinoid receptor genes and the fatty acid amyloid hydrolase gene, or Fa. So what we found, I already showed you this graph, a decrease in hippocampal volumes, but those with a genotype that puts one at risk for cannabis use shows even greater reduction in volumes in those regions. And in terms of response to cues, if you have, depending on the number of risk genotype, risk allele that you have, the greater response you have to the marijuana cues, okay, again, in the orbitofrontal cortex. So these are for the Fa and the CNR1 risk genes. So to conclude, marijuana users, that there are individual factors that increase susceptibility to marijuana's effects that include age of onset, stress, and also these genes that regulate the endocannabinoid system. But I'd like to point out that these are cross-sectional studies. We only collected data on one single time point. We didn't know how these individuals were prior to their marijuana use, so we don't really know if the group of users that we collected and which findings are related to may have already exhibited these effects prior to marijuana use. So we don't know if they have already been predisposed to these effects, and perhaps marijuana use didn't change any of that, or perhaps marijuana use exacerbated some of those, but we don't know. So a longitudinal study is what we need in order to address that question. Oops, excuse me. So with that, I'd like to thank you for your attention, and I think we have a few minutes for questions.
Thank you very much. There's certainly a lot of information for us to think about in terms of what factors may or may not predispose, particularly adolescents, to prolonged marijuana use. So we'll take questions. If we could use the two mics that are set up there so that we can record the questions as well as the answers. So we'll start with Bert.
On one of your first slides, I was really surprised to see the number of people seeking help for alcoholism and for marijuana abuse issues were, well, let's just call them almost identical. But the demographics were quite different. And it was children, essentially, 12 to 17-year-olds. And I wonder, are those people seeking help for a marijuana problem, or people with parents, oh my God, you're doing drugs, we're getting you counseling? Do we know what's going on here?
It wasn't specific. It could certainly be that the parents. So what we know for a fact that adolescents don't seek treatment on their own. So it probably is likely that parents being sensitive, right, being much more mindful of what their kids are doing. You're welcome.
Let's go ahead.
Hi. Was everyone smoking marijuana, or were any people eating cannabis?
Good question. Most, by and large, the population that we're drawing from only smoke. There's very little in terms of edible consumption in that population. We do collect information on how they use, and while it's diverse, there's very little in terms of edibles.
It looks as though for chronic marijuana smokers or cannabis smokers, there is a reduction in the concentration of dopamine in some brain areas. I was wondering if this perhaps means there's a connection between this and an increased risk of dopaminergic brain diseases like Parkinson's disease.
That's an interesting question. I don't know if I have to remind myself what that literature looks like, but the only I haven't seen that yet, and perhaps we just haven't waited long enough. These individuals haven't been followed over time yet. But what I have, and I failed to mention, but in terms of a dopamine disorder, what comes to mind is schizophrenia, and that is one that has been found to increase the likelihood of. So, but only again in terms of marijuana use in adolescents seems to, by some accounts, increase the risk of psychosis up to 3 times for adolescent use.
So I have a question regarding the study that you were doing regarding the object. So I think you would agree that perception is very subjective. So what if like if the subjects were shown like alcohol or any other illicit drugs, how would the reactions would be different from the marijuana as compared to other drugs or any other insensitive that they think is as valuable as marijuana? Because certainly I think everyone would agree that marijuana as compared to pencil wouldn't give me or anyone as much as insensitive to look at it, right?
Right.
Another question is that were there any anomalies in the data, for example, because since we are looking at perception, were there any anomalies like how people perceived fruits within like fruits? Were there like differences or were they like all same results that you achieved?
Same results in terms of their perception or awareness or
Of the object.
we didn't test that. So you're saying that perhaps there's some somatosensory effects that
Like because people have history regarding fruits, so how would you compare that with certain drug? So have you compared the drug with other drugs or any other incentives that they think are as similar to the drug experience?
I see. The answer is no. We haven't compared, besides food, we haven't compared anything else that might be equally salient. But that is an interesting question. Others have looked at not drugs, but similarly salient, provocative pictures, like one study that comes to mind compared cannabis-related images with sexually explicit images, and they found similar things where there's greater response to cannabis. Of course, it's not a drug, but in theory, it should elicit the same response.
Hi there.
Hi.
I just wanted to ask with regards to some of the MRI tests that you did. Correct me if I'm wrong, it was either marijuana users and non-marijuana users, but they were completely sober during the time of the testing. I was just curious if there was any testing that you maybe have done or somebody else has done using those MRI scans with someone who has consumed cannabis or taken cannabis. And if you can kind of look at the activity, like perhaps give someone a fruit that maybe has taken or hasn't taken and see if there's different activity in that sense, or if you can speak to that a little bit.
Let me make sure I understand your question. Is your question with regards to the control group, whether if we compared individuals to someone who wasn't completely naive, what that would look like?
I guess that as well, or even just with the
Within
cannabis
the
users,
users, okay.
maybe looking at how that brain activity differs
I see.
between cases.
I see. So in terms of your original question then, which is the last one you described, that would get at acute effects, right? So what would the acute effects what would the reward response look like immediately after somebody had just used? So that's a good question because we don't know, but also because since this was the first attempt at looking at response to cues in this group, we wanted to make sure that we got our biggest bang for our buck. And in fact, these users had been abstaining for 3 days. So imagine if you're a daily user, heavy daily, long-term user, and you have to abstain, your craving is at its peak following 3 days, right? And so that's probably why we're seeing these strong, robust effects. I bet only because behavioral studies have shown that following marijuana use, immediately following marijuana use, that effect wouldn't be so strong. And in fact, another way to get at that is when we look at expectancies. We've actually asked those questions. We asked them, do you plan to use marijuana right after this? And those that say yes seem to have greater response. So for those who have just been satiated, right, and their expectancies are low because they've already used, that would suggest that we would see a decrease or a reduced response.
Yeah, thank you very much.
You're welcome.
Dr. Filbey, thank you for your presentation. I'm aware that NIDA have spent a lot of time and money showing the risks and harms associated with cannabis, but they don't support trials showing the positive benefits of it. But I can assure you that the rest of the world is doing that. Do you see anything strange in the fact that at the same time the NIH, the National Institute of Health, hold a patent on cannabis as a neuroprotectant and antioxidant?
Yeah, so I mentioned to you the increased connectivity that we found, and we reported that in the literature. So that was one of the, I don't know if it's positive, but it's definitely not negative. It's not correlated with any, decrease in cognitive functioning, for instance. So we basically, we don't know what these effects really mean. They are certainly changes, but I don't think that we have enough to say whether they are negative or positive, at least from these studies, because they were not, again, related to any decline in function.
Okay, thank you.
You're welcome.
In your EEG study, you showed that marijuana users had, well, what looked to me like decreasing reaction times over the course of the experiment.
Yeah.
But you also showed that they had high alpha and pretty much flat light on beta. What was that compared to healthy controls?
We haven't looked at the healthy controls yet. That was as I mentioned, this is preliminary data and that we're beginning to look at. But there are a lot of studies on healthy controls using that task, the serial reaction time task. And although I haven't compared the users myself to a group of controls, controls decrease their behavioral performance, and it's the reverse. The literature shows that alpha is decreased and beta goes up and correlates with performance.
Sorry, I'll just a question here. Hi, thank you for your presentation. It was quite interesting how you're looking for the markers to seek with individuals who's looking for treatments and stuff, but are these effects that you see from the long-term usage of marijuana, is it specific to marijuana addiction, or is it some sort of a general trends that you see in other substance addiction, for example, alcohol? Like I was wondering maybe if you or any other person have done that, the experiment where you show the pipe and pencil, and same experiment with the alcohol and pencil or something like that.
Yeah, good question. In fact, I've done it in 3 other populations, in alcohol users, nicotine users, and also in binge eating individuals. And the findings are similar.
I was wondering if you did any differentiation between THC content and CBD content?
I'd love to do that experiment, but I haven't yet. I think that's an important one to conduct, but I haven't yet.
Hi. I crave apples and I crave using a nice sharp pencil, but I also crave cannabis at the end of a long, stressful day to help me sleep and relax. I can walk away from it easily. I don't need to consume it, but I enjoy it and I do. Do you think I have an addiction to cannabis based on that?
You would have to use the DSM criteria to diagnose a cannabis use disorder, but craving alone is not the only symptom. I showed a table that listed the symptoms, and you have to have demonstrated, 5 out of those 12 or 13 symptoms to do that. So craving is not the only symptom.
Hi. Do you have any plans for the future for other studies?
Yep. And actually, what we're doing now is we're looking at previous users to see if these effects that we're finding are also present in those individuals, or whether, to see if these effects persist or whether they normalize. I think that's an equally important question to ask. That's what I'm particularly excited about. But we're also looking at different ways that interventions could be improved. So we're looking at psychosocial interventions in those who are seeking treatment.
I was just curious about as dispensaries and everything have developed the more potent strains and the variations of sativas and indicas, have you looked at anything that pertains to chronic users that solely smoke more oils or like the more heavily concentrated THC forms?
Yeah, that is another good question. Unfortunately, we haven't done that yet because we cannot reliably, at least in my population, measure that. Two reasons. We aren't able to handle any of the substance ourselves to measure them, but also because participants, the population that we study in Texas and New Mexico, aren't very knowledgeable about the potency because it's not something that it's easily accessible to them.
Do you think the rise of dispensaries, like in Vancouver and places in Washington and Colorado, will allow them or that opportunity to study that? Like,
Yes.
That'll give you more options? Okay.
Yes.
Sweet, thank you.
Yeah, we'll do one more question and then we'll break for snacks and drinks.
So I know you're looking, and it's commendable, you're looking for risk factors and trying to find, however, I couldn't help but notice that all the studies, all the information you presented was strictly correlational.
Mm-hmm.
How can we make statements about whether something is detrimental if we don't know if the condition was pre-existing? I mean, it's the same thing with tobacco,
question.
right? But
exactly.
we don't have the mountain of evidence for marijuana as we do for tobacco.
I'm sorry?
And we don't have the mountain of evidence for marijuana that we have for tobacco.
Right, exactly. So yes, I think longitudinal prospective studies are the way to go. The problem with longitudinal studies is that we have to scan individuals before they use and then wait to see if any of them use. So that's an expensive proposition for me, although NIDA has done that with ABCD. But that's the only way to go, I think.
So she will be available here at the front if you have more questions. With that, I'd like to thank you for coming out and giving us a very interesting conversation.
Thank you very kind of you. I appreciate it.
Thank you very much.
You're welcome.